Hello, everyone, and welcome to today's live broadcast, Structure and Function of the Human Microbiome. I'm Bob Woodard, and I'll be your moderator for today's event. We are delighted to bring you this presentation presented by LabRoots. You can learn more about LabRoots by visiting www.labroots.com. Before we begin the presentation, I have a few important announcements to make. First of all, this is intended to be an interactive event. You are encouraged to submit questions and comments during the presentation. You can do that by clicking on the green Q&A button at the lower left of the presentation window and typing your question and comments into the Q&A box that appears on the screen. Also, if you'd like to enlarge the slide window during the presentation, click on the slide icon on the lower right of the slide window. And finally, if you have technical difficulties hearing or viewing this presentation, please click on the support button on the upper right of the presentation window, or let us know using the green Q&A button. And we'll try to resolve the situation as quickly as possible. Okay, that's it for that. Now I'd like to introduce today's speaker, Dr. Curtis Huttenhower. Dr. Huttenhower is an associate professor of computational biology and bioinformatics at the Harvard T.H. Chan School of Public Health and an associate member at the Broad Institute. He received his PhD from Princeton University, where he also performed his postdoctoral research at the Louis Sigler Institute. He was an analysis lead in the NIH Human Microbiome Project and currently co-leads the HMP2 Center for Characterizing the Gut Microbial Ecosystem in Inflammatory Bowel Disease. Dr. Huttenhower's lab focuses on computational methods for functional analysis of microbial communities. This includes systems biology reconstruction that integrates metagenomic, metatranscriptomic, and other microbial community omics, the human microbiome in autoimmune diseases such as IBD, and its potential as a diagnostic tool and point of therapeutic intervention. Please join me in welcoming Dr. Huttenhower to today's event. I will now turn the presentation over to him.
Great, thanks so much, Bob. Thanks to LabRoots for hosting today and to everyone for joining, especially anyone starting off the day bright and early from the West Coast. I'd like to provide a little bit of introduction today to the human microbiome. I'll talk about the ecology of the human microbiome, the NIH Human Microbiome Project and follow-up that we and others have been doing, and some of the analysis methods and tools that are used to study microbial communities. So one of the reasons that my group at the Harvard School of Public Health is excited about working on the human microbiome stems from the plot that you're looking at here. This is featured on the cover of one of our introductory biostatistics textbooks. And for those of you who didn't bring your magnifying glasses to the presentation today, I can use a slightly more modern version of this plot that shows deaths from infectious disease over the past couple of centuries, specifically in New York City. And if you follow the general contour of the plot, you can see that infectious disease caused by specific pathogens over the previous century was one of the leading causes of death in New York and, of course, globally. But then within the past century or so, advances in microbiology and in public health practice have reduced infectious disease, pathogen-driven disease, to a relatively minor source of mortality and morbidity in the developed world. Something that's really come to light in the past several decades, though, is the corresponding rise in non-pathogen-driven but microbially linked chronic disease. This is a widely used plot from a review several years ago now that shows the rise in a range of different autoimmune-linked diseases over the past several decades. Of course, much too quickly to be explained by human genetics, and instead speaking to changes in our interaction with our microbial environment. And of course, over the same span of time, several environmental factors that modulate our immune system's interaction with both typical and pathogenic microbes have changed as well, ranging from where we live and work, the types of foods that we eat, the antimicrobials and probiotics that we're exposed to, along with other pharmaceuticals, how we age, and even our day-to-day stress levels. And of course, the human microbiome in particular has been studied for quite a bit longer than that. Some of the earliest microscopy, like these illustrations that you're looking at here, was performed by Van Leeuwenhoek several hundred years ago, investigating under the very first microscope microbes from his own saliva. But the microbiome has become even more closely scrutinized in just the past few years due to one other plot that we can look at that features a dramatically changing line, which is, of course, the cost and throughput of nucleotide sequencing. So by taking advantage of the efficiency and effectiveness of modern high-throughput nucleotide sequencing methods, we can use DNA and increasingly RNA sequencing as a tool to get a better perspective on the human microbiome and how it might factor in these rapid changes in chronic autoimmune linked disease. So to establish some of the terminology that I and others will use over the course of the day, some of the earliest work in microbial communities occurred about 100 years ago now in the literature, at which point collections of microbes within a community were referred to as microflora. This is a term that you won't see very often anymore. Instead, you'll see things like microbial community or microbiota. And all of these terms, particularly microbiota now, are used to refer to a collection of organisms. And this typically spans not just bacteria, but viruses, eukaryotic microbes, such as fungi and archaea within a community. Fast forward about 75 years from there to the point at which high-throughput sequencing was beginning to take off, and the term metagenome was coined to refer specifically to the collection of DNA or the genomic potential of a microbial community. A few years later, the term microbiome finally showed up as well to refer to a community's total molecular repertoire or its local ecology. So you can think of the microbiome as the collection not only of specific organisms, but of their DNA, their RNA, their biomolecules, their metabolites, and their immediate surroundings, such that metagenomics as a field is overall the culture-independent study of microbial communities, be they from literally the environment, soil or water, or from a host-associated environment such as the human microbiome. So sequencing has become one of the main tools used to study culture-independent microbial communities. And to provide a brief overview of how this is performed, given a microbial sample, it's typically lysed by breaking open the cells, extracting nucleotides, most often DNA, but again, increasingly RNA to look at transcripts as well as genomes, and then performing one of two main types of sequencing. Either amplicon sequencing, in which PCR is used to amplify a single marker gene of interest. This is most often the 16S rRNA, which is a bacterially universal subunit of the ribosome, such that all of your sequencing throughput is used to sequence variants in a specific gene, variants at a specific locus. Each small nucleotide variant can then be used to classify that sequence to its microbial source, sort of like a microbial name tag. By counting how many times you see each sequencing variant at that one amplified locus, amplicon sequencing, or 16S sequencing as it's most often referred to, generates data that look like these, where for each sample, you get a list of which microbes are present and the counts or relative abundances at which they occur. Again, 16S RNA gene amplicons are most often used to target bacterial communities. Increasingly, you'll see the 18S RNA subunit, or ITS, internal transcribed spacer amplicons, used to target fungal eukaryotic microbial communities as well. Alternatively, metagenomic or metatranscriptomic sequencing just sequences everything. This results in a pile of short reads, much as you would sequence for a single genome isolate, and this can provide a more complete picture of what the total genomic potential of a community can look like, at the cost of a little bit more complexity and, as we'll see a little bit later on, a little more computational interpretation to help analyze the data. But as a result, you can see not just a taxonomic profile of which microbes are present, like you might from amplicon sequencing, but a functional profile of what, say, metabolism or pathways are present in the community, You can assemble contigs or increasingly near whole genomes from a metagenome, and this provides you an idea of which genomes, what gene content, and even if you sequence enough or have a simple enough community, what types of genetic variants might be present. And increasingly, especially in the context of public health, epidemiology-style microbiome experiments will integrate both of these types of sequencing along with other culture-independent or culture-based molecular tools to study microbial communities. This lets you answer a variety of different questions, often by slicing the same data set in slightly different ways. If you're interested in single specific organisms, you can zoom in on the sequences or sequence variants from a particular bug within a metagenome or other culture-independent sample. So for example, in the human microbiome, most of us carry slightly different variants of every organism. So we might all have slightly different E. colis. If you're interested in E. coli microbiology, you can zoom in on just the E. coli sequences from a collection of metagenomes. Alternatively, if you're interested not in any one particular organism, but in the microbial ecology or interactions within or among communities, you might zoom out a little and pay attention to which microbes occur together, how they might be competing or symbiotic within a community, or what their metabolic or signaling interactions might be. Zooming out still further, if you're studying the human microbiome, you might not care about the microbes at all, but use the sequencing readout as a high-throughput measure, say, of host health, much as we've become able to do using cancer transcriptomics as a high-dimensional diagnostic or prognostic readout. And finally, at the opposite extreme, if you're interested in public health or epidemiology, you might look at human ecology, or the interaction among multiple human hosts, or alternatively, environments. So this helps to address questions like how we acquire our microbiome in the first place after birth, or how microbial communities are transmitted horizontally later in life. All of these speak to different biological questions that can be asked with the same techniques, or often with the same data sets or collections of data sets, surveying the human microbiome or other microbial communities, again, in a culture-independent manner. One of the largest studies of the human microbiome to date has been the NIH HMP, or Human Microbiome Project, which had the mandate to establish what the baseline human microbiome looked like in the absence of perturbations from disease. The HMP ran originally from 2007 to 2012 and has been succeeded by a follow-up HMP2, or Integrative Human Microbiome Project, that I'll speak about in just a bit. The HMP itself, though, to establish a baseline of our normal microbial variation, recruited a population of 300 healthy subjects, all half adult men and half adult women, because unlike our genomes, our normal microbial populations vary a great deal from person to person. These individuals were surveyed at up to 3 different time points each, at up to 18 different body sites grouped into 5 areas shown on the upper right: the nasal communities, several different oral communities, several skin microbial communities, the gut as represented by a stool sample, and multiple different vaginal sites in women. These samples were accompanied by clinical metadata, demographics, biometrics on these individuals, and all of the sequencing data, both amplicon and shotgun metagenomes, are available at the Data Analysis and Coordinating Center shown in the lower left. The original series of HMP publications included between 5,000 and 6,000 amplicon sequenced data sets, if you multiply out individuals times time points times body sites, and about 700 metagenomes, surveying a subset of individuals, time points, and body sites. We've subsequently sequenced another 500 or so metagenomes, resulting in a collection of over 1,200 metagenomes, surveying 100 individuals at almost all 3 time points each. Again, all of these first and second wave data are available at the Data Analysis and Coordinating Center, and there have been a series of publications released from the HMP summarized at the Nature collection linked on the slide. To provide an overview of some of the consortium's early results, however, we can zoom way out and look at just the very basic outline of what this survey of microbial diversity told us about the human microbiome. What you're looking at on the left here is a summary of the 18 body sites surveyed in the HMP, connected by lines indicating how similar the overall microbial communities or beta diversity of these body sites was on average across the whole population. So body sites that tended to contain the same types and abundances of microbes are connected by stronger lines. You'll see that this groups the microbial communities into several different clusters by biogeography, including our several oral sites on the right, which grouped into 3 main collections: the hard surfaces of the tooth just above and below the gum line on the lower right middle, several different frontal and distal soft tissues on the upper right, and then at the other end of the GI tract, the gut microbiome is quite distinct and has been perhaps the most closely studied area of the human microbiome. We'll speak to that a little bit later on, since it's by far the region in which we carry the greatest microbial load, and it represents the greatest exposure of our immune system to commensal microbes. On the upper left, we have several different skin sites. As you'll see in the next few slides, these tended to be the most variable both over time and among individuals, most likely because they're more exposed to the environment. It's easy to, for example, wash our hands and change which microbes we have on our skin. And then in the lower left, the 3 different vaginal communities, the 3 different vaginal body sites surveyed for female subjects tended to be very similar to each other in terms of microbial ecology, and we'll see why that is in a few slides as well. We can expand these summarized relationships, however, and fill in a little bit of the population density of this broad outline. What you're looking at here is an ordination of all 5,000 or so amplicon datasets generated during the first wave of the HMP, such that now each point represents just one sample, and they're closer to each other if their overall microbial communities are more similar. So this helps to expand out the summary that we're seeing on the left, and you can see that of the many different oral body sites surveyed, even though there's diversity throughout the oral community at different locations, they're generally more similar to each other than to different body sites. And then on the left, there's a range of microbial diversity from the skin and nasal sites, which were somewhat similar to each other. The very front of the nostrils was sampled during the HMP, so it looks very much like a skin site, through the vaginal communities to the, again, very distinct gut communities on the top middle. To continue zooming into a little bit more of the ecology of these samples, terms that you'll probably hear several times today include alpha and beta diversity. Alpha diversity in microbial ecology represents the number of different organisms within a community and how evenly they're distributed. So if you have many different organisms and they are comparably abundant to each other, you can refer to that community as highly alpha diverse. Similarly, if you have just a few organisms or just a few organisms that are more abundant, that community is less alpha diverse. So what you're looking at here is a summary of the alpha diversity or within community diversity across the 18 body sites of the HMP. And the box plots, the vertical range of each box, summarizes the span of alpha diversity across the 300 individuals in this healthy population. The 3 different colors represent both our 2 different data types, amplicon or 16S sequencing, and metagenomic or whole genome shotgun, WGS as it's summarized here, metagenomic sequencing. The amplicon sequencing was processed in 2 slightly different ways computationally to understand either direct classification of these sequences or phylotypes, or cluster-based classification of these sequences, or operational taxonomic units, OTUs. Fortunately, both of these data types and all 3 different ways of processing them tend to agree with each other very closely when surveying the alpha diversity of our 18 different body sites. You'll see in the middle of the slide, the gastrointestinal tract, including both the gut and the oral community, tended to be more alpha diverse. We tend to have more bugs at these microbially accessible mucosal nutrient-rich sites. And then areas like the skin were again less alpha diverse. And in this case, for reasons we'll again see in a few slides, the vaginal microbiome was particularly low diversity. As we'll see a little bit later, the vaginal microbiome tends to be dominated in many individuals by just a few organisms. So again, that's reflected in a lower alpha diversity. The next type of microbial diversity that's quantified in microbial ecology is beta diversity, or between community diversity. So beta diversity represents how similar or different two separate microbial communities are. So if individuals tend to have mostly the same organisms at the same abundances between the two different communities, then we'll say that they are less beta diverse. If those two communities tend to be more different, we'll say that they're more beta diverse. So beta diversity acts like a distance or a correlation measure. It tells you how similar or different two communities are rather than summarizing the properties of one community the way that alpha diversity does. So for example, on the left side of this slide, you can see that the differences between skin communities among individuals tend to be pretty high. So like I said earlier, the skin community is a little easier to disrupt, it's a little sparser, and so when you look from person to person, you'll often see different organisms, thus resulting, again, no matter how the data are processed, in a higher beta diversity on the skin. This is less true in the gastrointestinal tract, even though we tend to have more bugs in our GI tract, the oral and gut communities. They're more often similar, although as we'll see in a few slides, more similar still means quite different from person to person. And then finally, on the right-hand side here, you'll see an interesting property of the vaginal community. If we look at our amplicon or 16S data using direct classification or phylotypes, these represent approximately a genus-level classification of the sequences. And there are relatively few differences among the genera of microbes carried in the vaginal community. So the red boxes on the right tend to be very low. However, if we use operational taxonomic units, or OTUs, that cluster our amplicon sequences to a slightly higher level of specificity, we start to see more differences. It turns out that in the vaginal community, many individuals, not all, but many, tend to carry predominantly microbes from the genus Lactobacillus, but they'll often be different species from within that genus, such that if we classify those sequences more precisely in the yellow boxes, we see more differences between individuals. And then finally, using our shotgun data to get a very precise level of taxonomic classification, the blue box on the right-hand side is even higher. So this speaks to the different ways you can slice the same data sets like I referred to earlier. If we ask the question, do individuals tend to carry the same microbial genera in the vaginal community, the answer is yes, and they're less beta diverse. If we ask the question, do individuals tend to carry the same strains of microbes within the vaginal community, the answer is no, and our blue box shows more beta diversity. So we'll see different ways of analyzing microbial community data over the course of the talk. I want to speak to this a little bit by describing some of our pipelines for analyzing shotgun metagenome and metatranscriptome data. And several of these are built on a collection of tools referred to as the FLAN tools, or phylogenetic analysis, originally started by Nicholas Segato when he was a postdoc with the group. He has his own lab at the University of Trento now, where he's continued a series of computational methods development for assembly-free metagenome and metatranscriptome analysis. This relies on the intuition that we can use microbial isolate genome information to analyze microbial community metagenome information. So Nicholas started with two sets of data, referred to here as RepoPlan and ChocoPlan, built on the total collection of microbial isolate genomes currently available at the NCBI. This includes not just bacteria, but again, archaea, viruses, and eukaryotic microbes, primarily fungi, and spans both draft and complete genomes, totaling about 20,000 genomes right now. We're getting on the order of 1,000 new microbial genomes per month. So this is a tremendously powerful tool for turning those isolate genomes into information about communities. RepoPlan is a system that downloads these genomes, organizes them, performs some quality control and filtering and taxonomic reclassification, and then identifies the protein coding and other open reading frame genes in this collection, which is currently just about 50 million open reading frames or ORFs. These are then cataloged by a second system referred to as Chacoflan, or the catalog of homologous clusters of gene families, which extracts the open reading frame collections from each species to create a pan-genome. This currently spans just over 7,600 species and totals about 20 million gene family clusters. These are then annotated with phylogenetic and functional information, including which genes are core, that is, conserved throughout a particular species or clade, and which of those core genes are unique markers. The unique marker genes are among the most taxonomically informative because they are carried by all members of a species or clade, and they are not carried by other organisms. So each time we see a sequence from these unique marker genes in particular, we know which organism it came from. And this provides one way to turn shotgun metagenomic information into a taxonomic profile, counting up which bugs are there, just like you might with amplicon sequencing data. Nicola put this together in a tool called Metaplan, and what you're looking at here is a taxonomic survey of the first 700 metagenomes from the human microbiome using Metaplan. Each column represents one metagenome. The body sites from which they're drawn are annotated along the top of the slide in color, and each row represents the relative abundance of one organism across those 700 samples. And this represents just a slice off the top of the most abundant organisms throughout the human microbiome. Going from left to right, if we just naively cluster these, you'll see that the major driver of microbial composition in the human microbiome is biogeography, is the body site from which that community is drawn. In the upper left, orange represents the nasal communities with a few other skin communities mixed in. The next three colors are from the oral microbiome, respectively green from the tongue, red from the cheek, and brown from the tooth surface. And again, while these are quite distinct from each other, you'll see that they're more like each other than they are other body sites, just as we saw earlier. Purple represents the gut, and blue on the lower right represents the vaginal microbiomes. Now, I want to zoom in on an example from the gut and vaginal communities in particular. Since a few years ago, a collection of high-profile publications, originally in Nature and later picked up by other scientific journals such as the New York Times, raised the hypothesis that we could organize the complexity of microbial ecology in the gut into just a few discrete types. And to provide an example of what this might look like, the vaginal community does appear to be organized this way. If we zoom in on the 50 or so metagenomes from the posterior fornix, one of the vaginal sites surveyed by the HMP, and we look at just the abundant species in these communities, you'll see that there are 4 clusters, each dominated by a particular lactobacillus species. So going from left to right here, we have Lactobacillus crispatus, Lactobacillus jensenii, Lactobacillus eners, and Lactobacillus gasseriae. There's a 5th cluster over on the right-hand side, represented by just a few individuals in this population, that lacks any lactobacilli and instead has a diversity of other organisms. These 5 quite distinct community types have occurred pretty reliably in the vaginal microbiome across different populations. And in each one, either you're dominated by a particular lactobacillus species and lack the others, or you lack all lactobacillus species. So this organizes the complexity of the vaginal ecology into 5 main community types. The hypothesis was that you could do something similar for the gut, but you can see even by eye that this type of microbial organization doesn't seem to appear in the gut. And if we zoom in to a higher level of detail in both the HMP gut metagenomes and a collection of similar metagenomes from the MedHit project, which is a parallel European project, we'll see that there doesn't seem to be that type of discrete clustered organization. There's a great deal of diversity, differences in which microbes are abundant from individual to individual, and there are certainly microbes that are quite abundant in several different individuals. But if we ordinate these samples, again, this is a visualization in which communities are projected into a 2-dimensional plane. Each point represents one microbial community. And the more similar they are, the closer they appear on the ordination. You'll see that there don't appear to be discrete clusters, except in one case, and we'll talk about that in just a second. Instead, there's a continuum, a smear of communities ranging from those that tend to carry most members of the Bacteroides, or more generally the phylum Bacteroides, one of the two main groups of microbes in the gut, down to individuals that carry relatively few Bacteroides and mostly members of the phylum Firmicutes instead. The exception is this outgroup here, which are individuals that tend to carry specifically members of the genus Prevotella. So this is something that occurs pretty reliably across Western populations. Other parts of the world are a little bit different in terms of gut microbial ecology. But in this particular case, we have a subset of about 10% of the population that looks more like the vaginal microbiome organization. If you have Prevotella, and in this case specifically Prevotella copri, it's abundant and you're depleted for other organisms. This results, if you quantify the cluster structure in the data, in 2 reliable clusters in the gut microbiome, Prevotella and everything else, as opposed to the 5 very reliable clusters we saw in the vaginal microbiome. And this is work in quantifying this intuition rather than just relying on our eyeballs. It was carried out by Levi Waldron and my group in a collaboration with Ruth Lay and Rob Knight's group. Interestingly, in this particular example, we do reliably see this Prevotella outgroup in other populations, and we've since gone on and worked with Dan Littman to show that it's associated with new onset untreated rheumatoid arthritis. So this is an ordination of a completely separate population that Dan recruited in New York, in which we have the same gradient from high to low Bacteroides carriers, the same outgroup of Prevotella carriers, and that outgroup is enriched for new onset rheumatoid arthritis patients. We don't yet know the causality. We don't know if you're at greater risk of RA due to Prevotella carriage, or if the onset of RA increases Prevotella carriage. But we're going on to do some mouse experiments with Dan to try to tease that apart. Continuing to zoom in, however, we've looked so far at species-level taxonomic profiles from metagenomes using Metaflan. We recently completed a second version of the tool, Metaflan 2, that includes not just bacterial and archaeal sequences, but also viruses and eukaryotes, and which allows you to zoom all the way into subspecies or often strain-level microbial profiling. This is an example from a collection of skin metagenomes into which we've zoomed in a little more detail here. If you're familiar with skin microbiology, you'll see several of the usual bacterial suspects, but also scattered throughout here, examples like Malassezia gibosa towards the middle of the plot, which is a common skin fungal microbe. And down towards the bottom, several viruses, specifically Propionibacterium phage, and different variants of that phage that occur in Propionibacterium hosts in different human hosts. If we continue to zoom in to this type of taxonomic profile, we can actually identify different strains of microbes carried stably by different individuals. What you're looking at here is an example from the vaginal microbiome, in which the unique marker genes for Lactobacillus crispatus have been unrolled. So from left to right in this plot, you're looking at several hundred different loci across the Lactobacillus crispatus genome that we have reason to believe from isolate genomes come specifically from Lactobacillus crispatus. We don't see these sequences in other microbes. However, not all Lactobacillus crispatus strains carry exactly the same genes. Microbial genomes are much more plastic than our own, such that in these 2 individuals, with 3 time points represented in the top individual and 2 time points represented in the bottom individual, we can see slight variations in which genomic loci are present both between these individuals' Lactobacillus crispatus strains and between these strains and the reference pangenome that we have for Lactobacillus crispatus. So several of the marker loci that differ between these 2 strains have been indicated by red arrows here. And you can see that for the most part, over these time points spanning about 6 months of difference each, excuse me, the same strain is carried by each individual. There's one change in the lower individual in which one of the genomic loci, which looks to be a mobile element, has been picked up and modified such that the strain changes very slightly. But for the most part, not only in the vaginal microbiome, But across different strains of different microbes at different body sites, this is a general property of the human microbiome. We all have slightly different strains of most organisms, and in our adult life, over moderate periods of, say, months to a few years, these strains tend to be pretty stable. This is interesting in as much as a postdoc in my group, now a research scientist, Eric Franzosa, was able to quantify the degree to which our microbes really are personalized and unique. So in work with Brendan Bohannon at the University of Oregon and Catherine Lemon at the Forsyth Institute here in Boston, Eric asked the question, can we construct sets of features that uniquely and stably separate different individuals, identify different individuals, based not on our DNA, but on our microbial DNA? And you can think of features as including either specific microbes, which bugs we carry, microbial genes, like we just saw, which loci are present in our microbial strains, or microbial variants, individual polymorphisms. And you need a lot of sequencing to get that type of information from a whole community. So any such set of features we'll refer to as a metaomic fingerprint. You can combine microbes or microbial genes or microbial SNPs to try to separate out, to uniquely identify individuals in a population. So I won't go into the algorithm that Eric used to construct metaomic fingerprints, but I'll provide a brief overview of the results here, either using taxa on the left from the amplicon or 16S data in the Human Microbiome Project, taxonomic profiles at the species level in the middle from our shotgun metagenomes, or combinations of marker genes like we just saw on the right-hand side. And you'll see that if you try to separate individuals just by which bugs they have at the OTU or species level, often we can't uniquely separate individuals. The alphabet, or the number of different bugs, is a little too small. But on the right-hand side, if we consider which genes are carried in combination by our microbes, we're often able to construct codes which separate out individuals, and comparing the blue versus red bars on the right-hand side, those codes are often stable over time. So what we're looking for here are red bars that represent true positive identification of individuals using metaomic fingerprints, made in this case from marker gene combinations, that are stable over multiple time points. So these are represented by red bars. If we lose an individual, we say, here's a code for somebody, but it's gone at the next time point, that's represented by a blue bar. And then false positives, in which a code misidentifies the wrong individual, are shown in cyan and purple on the right-hand side. Unfortunately, those bars are small. So most often, ranging from about a third of the time in the trickier body sites like the skin, up to about 75 to 80% of the time in a more stable body site like the gut, we can construct a code based on strain-level identification of combinations of marker genes that uniquely identifies an individual and, as is shown across the bottom here, is stable over time. There's relatively little effect of time intervals on the order of weeks to months to a year or two in these particular data on the ability of these codes to uniquely separate out individuals. So just like we can use variants in our own DNA to tell one person from another, to a degree, we can use variants in our microbial DNA to tell one individual from another. So this helps to start explain why our microbiomes are so distinct from each other. If we acquire different organisms and they're relatively stable over time and settle into different patterns of strains that we each carry for, say, months to years over the course of our adult life, that helps to create that higher degree of beta diversity that we saw earlier. However, something that's common among individuals in the human microbiome is the functional repertoire needed to maintain a microbial community at each environment. So what you're looking at here on the left-hand side, on the top is the differences at a high level, at the phylum level, from individual to individual in which microbes we carry at different body sites. However, in each of these same samples, what you're looking at on the lower left is a collection of genes and pathways carried by the same microbial communities at each body site, at each sample. And so if we look at something like the gut, blue represents Bacteroidetes. So again, we see this continuum from individuals with a whole bunch of Bacteroidetes down to individuals with almost none. But the pathways on the lower half of the plot needed to maintain a healthy gut microbial community are relatively stable. Certainly different individuals will carry bugs with different genes, but the overall metabolism and signaling and regulation needed for that community is relatively stable, even though it's carried by different bugs in different individuals. This property has been observed in other cohorts. It's even been observed in other hosts. This is a great paper, except that it's not about the human microbiome at all. It's about algae-associated microbial communities. And again, this ability to distribute responsibility, to distribute different metabolic or regulatory needs among organisms, is in part explained by mechanisms of plasticity like horizontal transfer that are much more available to microbes than they are to typical eukaryotic hosts. So in a way, this distribution of genes and pathways is the core human microbiome, not necessarily a distribution of particular organisms. And although I won't go into it much today, it's amazing how much of this is microbial dark matter, how many of these sequences are not yet functionally characterized. We don't necessarily yet know what they do or how they get there or how they're maintained over these longer periods of time. But there's a lot that we do know about the microbial metabolism and regulation in the human microbiome and other communities. I'll close over the next several minutes with a couple of functional profiling tools and examples that we and others have developed. One example that I'll speak to briefly is called a shortbread, or the short better read dataset assembler. This is a functional profiling tool that uses the same intuition that Metaflan does, in as much as we can identify unique sequences from clustered protein families of interest. So if you have just a few types of sequences that you're interested in picking out of a metagenome or metatranscriptome, shortbread will take that list of sequences, cluster them into homologous families, identify just the unique regions that separate each family from others and from other proteins, and then count up how often those unique sequences occur in a metagenome or metatranscriptome. So this is developed by a former master student with the lab, Jim Kaminsky, now in the Berkeley PhD program, and has the benefit of being for specific gene families of interest, faster and more specific. So just like Metaflan focuses on just the sequences that are taxonomically informative, shortbread focuses on just the subsequences that are functionally informative for particular protein families of interest. So these might include, for example, antibiotic resistance sequences. This is an example of shortbread profiling that we did with Gautam Dantas' lab and his student Molly Gibson at Washington University, combining 3 different gut metagenome cohorts from the HMP, the global gut survey carried out by Jeff Gordon's lab, and the survey of type 2 diabetes subjects in Chinese gut metagenomes, identifying antibiotic resistance sequences from the ARDB, or antibiotic resistance database in this case. And you can see that there's both some consistency across these global gut metagenomes, including tetracycline resistance occurring in high abundance and high prevalence in nearly every gut metagenome, down to variants that occur only in, for example, the Chinese subset of the population. And you'll see a little bit more about this later in the day for antimicrobial resistance profiling. However, if you're interested in broader functional profiling of metagenomes or metatranscriptomes, the tool that we've developed for the HMP, referred to as Human, or in this case, the second version of that tool, Human 2, provides a broad functional profiling of any gene families from human or other microbial communities, both metagenomes and metatranscriptomes. Human is built on the same collection of genomes and genes that we saw earlier, Riboflan and Chocoflan, that drive Metaflan 2. However, instead of, again, using just the most taxonomically informative sequences like Metaflan 2 does, Human 2 uses all functionally characterized sequences from this catalog and will efficiently profile the genes and pathways in a metagenome or metatranscriptome by starting first with a survey of the most abundant microbes in that metagenomic or metatranscriptomic sample. By lifting the pangenomes from those abundant organisms out of Chocoflan, we can rapidly map nucleotide sequences to them using Bowtie2 to identify the gene families and bugs that they're drawn from. For sequences that don't hit, for reads that don't hit using nucleotide mapping, HUMON2 next performs a more general translated search using DIAMOND, which is an accelerated translated BLAST tool. And finally, hits at either the nucleotide or protein level are reassembled into pathways using Metacyc by default, but HUMON2 can also use other pathway databases such as Unipathways or KEGG if those are of interest. So this results in a list of which gene families are present in a sample, their relative abundances, and when they can be identified, which microbes they're drawn from. These gene families are also reassembled into pathways. So just like we typically get a taxonomic profile from amplicon data or from metagenomes using a tool like MetaFlan2, HUMON2 provides a functional profile of either gene or pathway relative abundances or counts. We can use that in contexts like the study of inflammatory bowel disease, getting back to the original motivation for looking at the human microbiome and its association with complex chronic autoimmune disease. IBD, inflammatory bowel disease, has become one of the poster children for complex involvement of the microbiome in chronic disease over the past few years, starting with the observation that the gut microbiome was involved somehow a few years ago, some general observations of disruption or dysbiosis in microbial ecology, and then over the subsequent few years, refining that into more and more precise observation of which microbes were enriched or depleted during the disease, and how these varied among patient subsets, particularly between the two major subsets of inflammatory bowel disease, Crohn's disease, which you'll see referred to as CD here, or ulcerative colitis, which you'll see abbreviated as UC. And I like to think of the involvement of the microbiome in disease very much like the involvement of human genetics, in as much as certain diseases are genetically Mendelian, cystic fibrosis can be blamed on one particular locus, and certain infectious diseases are, certain diseases are infectious, are able to be blamed on a specific pathogen. Other diseases are genetically complex, such as, say, type 2 diabetes, and other diseases are microbially complex, such as inflammatory bowel disease, in as much as it can't be blamed on one particular organism or pathogen. So to help understand this complex involvement of multiple microbes or the whole gut community in IBD, we wanted to ask the question, which of these overall ecological structural changes might be functional, and why? How might they associate with specific microbial metabolic functions in specific subsets of IBD? So we originally started looking at this question a few years ago, driven by a research scientist in my lab, Sochi Morgan, and a collaboration with Ramnik Xavier at Mass General and the Broad, and Bruce Sands in New York. The first set of cohorts that we looked at was a combination of two cohorts from these locations, Oscar and Prism, totaling just over 200 individuals, from whom we had a good bit of, again, accompanying metadata and clinical background on their disease progression and on treatments that they had been exposed to. We surveyed these individuals using two different types of samples of the gut microbiome, both stool samples and mucosal biopsies. And if you look in the background of the slide, you can see that, again, the ordination of microbial similarity between these 200 individuals kind of separates out ulcerative colitis and Crohn's disease a little bit from our healthy controls. But just disease, IBD subset is not the major driver of microbial diversity in these individuals. And as we expand out and overlay the other environmental exposures that help to explain this microbial variation, you can see that there's a whole range of things that affect the microbiome. And this is a general principle. In as much as things don't affect our genetics, lots of things affect our microbial communities. These range from how we survey, what type of sample we use to look at the gut microbiome, ranging from mostly stool samples on the right to mostly biopsies on the left. Treatment exposures, so for example, in this cohort, UC patients more often received antibiotics, and we can identify an independent effect of antibiotics on the gut microbiome that's different from what ulcerative colitis alone has. And then similarly on the left, Crohn's disease patients more often received immunosuppressives, and those had another different effect on the gut microbiome. Finally, along the diagonal axis, Although it might be interesting to look at the effects of smoking on the gut microbiome. In this case, a much larger effect was age. We had subjects ranging from pediatric patients up through older adults. And the microbiome does vary over time, particularly during infancy and childhood, such that what we're seeing in that axis of microbial variation is, we think, not smoking, but the fact that most of our healthy controls were, there was an enrichment of our healthy controls for pediatric subjects, which is great, and they were mostly non-smokers, which is also great, such that we see microbial variation due to age not smoking. But what we'd like to do is tease apart just the effects of disease on the gut microbiome. So using a multivariate analysis, we did that both for microbial taxonomic profiles and for microbial functional profiles. And this is one of the most important conclusions here, inasmuch as IBD, again, appears to be not a function of specific microbes, but of specific microbial processes. Just like what is maintained in the gut and other microbial communities during health is not specific organisms, but a distribution of necessary functions. What's disrupted in IBD is not necessarily specific bugs, but a set of microbial processes that become dysbiotic. In this case, proportionally over 6 times as many as specific organisms. And so the parallel that I would draw here is in terms of a city's ecology. If there's a problem with the transit system, it's not because of people with a particular name, but of people with a particular job. If there's a dysbiosis in the gut during IBD, it's because of disruptions to microbial processes, in this case, often the response to redox stress induced by inflammation and activation of the immune system. It's concentrated in particular organisms. So the Enterobacteriaceae, for example, are often enriched during chronic inflammation, such as IBD. But the driving factor is selection on that process, not necessarily a pathogen or specific organism in the traditional sense. We're continuing work on the IBD microbiome now during the HMP2, or Integrative Human Microbiome Project, a follow-up to the first wave of the HMP. And this is an ongoing study, again with Crohn Xavier at the Broad and MGH, in which we're following 90 individuals drawn from a variety of IBD phenotypes for 1 year each, and longitudinally surveying their gut microbiomes through a variety of different modalities. Part of the mandate of the HMP2 is to provide multi-omic data that really serves a comprehensive survey of the microbiome and host molecular responses in different diseases. Our project looks at IBD. There are several other HMP2 projects looking at other conditions such as type 2 diabetes. And if you're interested in more information there, you can check out the IBD MDB or IBD Multi-omics database site on the slide here. The project's still ongoing, but to wrap up with an example, we have a pilot data set in which we've surveyed about 200 gut metagenomes from individuals in this population, accompanied by stool metabolite profiles, so stool metabolomics. We can then ask the question, which microbes are differentially abundant during Crohn's or colitis, and which metabolites are differentially abundant during Crohn's or colitis, and start associating compounds with bugs to help explain at the molecular level why these organisms and possibly mediating compounds might be disrupted in disease. So what you're looking at here is a correlation on rows between bugs and on columns between metabolites that are associated with each other over and above the amount that we'd expect just from the disease itself. So this starts to suggest that these compounds might be associated with these bugs for mechanistic reasons. The bug might produce that compound or consume that compound, and we're currently doing some genome gazing to help tease apart those potential causes. And one example that we can obtain using a specific tool like HUMON2 is to survey essentially positive control compounds. In this case, the bile acids are known to be disrupted in IBD. Different members of the family of bile acids are either enriched or depleted during Crohn's and colitis. These are known microbially processed compounds that are generated by the human host and then processed by microbes in the gut. And what we're looking at here is a subset of our 200 metagenomes in which each row picks out one gene family that's a member of the bile acid hydrolases, which is the first enzyme in the microbial pathway that processes the bile acids. Each row represents one member of the bile acid hydrolases drawn from one specific organism. If you squint real hard, you can see a gene family identifier and a microbial taxon associated with each row. A subset of these specific gene families are again enriched or depleted during Crohn's and colitis. And this allows us to make, from high-throughput data, very specific hypotheses that say these bugs are disrupted in this phenotype due to changes in these particular selective pressures associated with these particular pathways, and in this case, these specific gene families. So this is one way in which we can tie together a very specific mechanistic molecular hypothesis about how and why microbial ecology might be disrupted at a functional level during a complex condition such as inflammatory bowel disease. I'll wrap up by pointing out that the tools used in these examples and several others are available on the lab's website, or the BioBakery, which is a collection of documentation, downloadable software, online tools, manuscripts when available, source code, hopefully everything you need to run this family of tools for metagenome and metatranscriptome analysis on your own data. And again, emphasizing that in as much as there's a lot that we do know about the human microbiome so far, there's a lot that's still left to learn, ranging from basic microbiology and mechanism all the way up to more complex human or microbial ecology. This includes, as I mentioned earlier, a whole lot of functional biochemical dark matter sequences that are not yet functionally characterized either in the bugs or on the host side, particularly in terms of host microbe immune interactions. As far as translating the microbiome, again, we have opportunities not just in diagnostics to use the microbiome as a high-throughput readout of health, but as intervention. It's hard to change our genomes, but it's surprisingly easy to change our bugs. It's just really hard to do it in a way that does exactly what we want or induces the phenotype that we'd like. And finally, on the public health and ethics side, there are questions of, for example, microbial privacy, as might be raised by the identifiability study that I mentioned earlier, or in health practices. There's a lot of great work in the field that I haven't described today on early life exposures and how perturbations like antibiotics might induce changes in our microbiome that are linked to health status later on in life. So with that, I'll wrap up. I'll thank again the members of my own lab who did the hard work on all of these projects on the left, our great collaborators in the HMP and the HMP2 on the right, particularly Ron McSavior and Dirk Beavers, who were involved in pretty much all of these examples from beginning to end, Wendy Garrett at the Harvard School of Public Health, and I'm happy to take any questions today. Thanks, everyone. Wendy Garrett at the
Well, thank you, Dr. Huttenhower, for that informative presentation. Before we begin the Q&A session, I'd like to remind everyone how to submit their questions and comments. You can do so by clicking on the green Q&A button in the lower left of the presentation window and typing them into the Q&A box that appears on the screen. Our first question comes from Manesh Sriramulu of Trust Consultancy, India, and Ataturk University. His question is, what is the level of overall stability or representativeness of human microbiome in normal individuals?
So the work that I'm familiar with that has assessed microbiome stability in the most detail comes from Jeff Gordon's lab and looks specifically at the gut. They asked the question, what's the decay rate for retention or loss of microbes over time? And what you see is a power law distribution in which microbes that are going to be lost are lost fairly rapidly. After the first week or two subsequent to sampling, most microbes that made it that far tended to stick around for months to years. Both their survey and the data that we've looked at during the Human Microbiome Project ranged out to a couple of years at most, over which time there was a surprising amount of stability. And in other data that we've looked at, you can actually see effects of childhood exposures in adult life. So for example, breastfeeding or mode of delivery, vaginal versus C-section, does leave microbial echoes that you can see in the microbiome 20, 30, 40 years later on. So as far as specific microbes, the dynamics tend to be on the order of weeks to years. as far as the overall microbial community, you can see configurations, effects of exposures that last certainly years and possibly decades as well.
Thank you. Our next question comes from Togba Onal Okeye, University of Houston. His question is actually, I think, refers to slide 11 in your presentation. He's asking if you could repeat how to read this graph.
Great. I am trying to get us back to slide 11 in case that will display as well. There we go. So in both this slide and the previous slide on alpha diversity, this slide represents beta diversity, or differences between microbial communities. Each of the columns along the x-axis represents one of the 18 body sites surveyed by the HMP. Each box represents the beta diversity across the population, or the aggregate of all pairwise differences between individuals at that body site. The three different colors represent three different ways of processing the HMP data. Either our amplicon data, directly classified as phylotypes, or clustered as OTUs, or our shotgun metagenomes, referred to as WGS, or whole genome shotgun data in this particular plot, classified to reference genomes. So each box represents the beta diversity, differences between bugs, between individuals, across the whole population at one body site. Higher means more differences, and lower means fewer differences.
Well, thank you. The next question is from Fernando Horato Ange at Laval University. Why is, excuse my pronunciation here, why Microbia is often not found in these analyses if it is such an abundant phyla in the gut microbiota?
I'm trying to jump back. There we go, to the closing slide. If I heard correctly, Verruca microbia is among the clades that can be a little more difficult to detect, either due to 16S primer specificity. So when using amplicon data, one of the potential biases is in the different affinity of specific primers for subregions or hypervariable regions of the 16S RNA gene, different affinity in different organisms. So even though many primers that target different regions of the 16S gene are referred to as universal among bacteria, they tend to match slightly better or worse to different organisms. Verruca microbia can be underrepresented if the primers don't match their particular 16S variants quite as well. Another great example is in one of the most common primer sets used right now that targets the V4 hypervariable region of the 16S. We'll almost never see Propionibacterium, which is one of the more common skin microbes. Alternatively, on the shotgun metagenome side, one of the reasons that some clades, including sometimes the Verruca microbia, are underrepresented is a dearth of reference information. This is even more common in unusual clades that aren't well represented by reference genome information. So if you're not performing de novo assembly of abundant microbes in metagenomes, it can be hard to see bugs for which we don't have enough reference information on the shotgun side. So depending on your sequencing type, those are two reasons why the Verruca microbia, and again, often other more unusual clades, can be underrepresented.
The next question is from Jovan Trapark, Faculty of Biology at the University of Belgrade. This question is about this biogeography research of the microbiome. Have they observed like male genital microcommunity?
I apologize that I didn't quite hear the tail end of the question. There have been studies of both the urethral and the general male genital microbiome as well. I'm not personally familiar with them, but I know there are several that have been published in the literature.
Thank you. Here's another question from Fernando Ferrato Ange, Laval University. What's your opinion concerning the use of the 16S rRNA-based taxonomic profiles to predict community function by applying algorithms? And wouldn't that be very limited, as it assumes that different strains of the same phylotype exert the same functions?
Great. This question refers indirectly to a tool that we built with Rob Knight and Rob Byko, referred to as PyCrust, which will perform an approximate functional inference or metagenome inference from a taxonomic profile. So the brief summary is that given a taxonomic profile of which bugs are present, typically from amplicon sequencing, by combining that with knowledge of which genes those bugs typically carry from reference genomes, you can make an informed guess about which genes and thus what the metagenome looks like for a community. You're exactly right in as much as that is an approximation. PyCrust uses ancestral state reconstruction to make a quantified guess about which genes each bug probably carries. That comes with confidence intervals based on how often each gene is observed in reference sequences to be present in each isolate genome. There are some genes that are always carried by a particular clade. There are some genomes that are variable as part of the pan rather than core genome. So PyCrust does the best it can. It will provide you with confidence intervals indicating which genes are less certain to be in the community. And it does assume that different strains within a particular clade are equivalent in some sense to the degree that they can be resolved by 16S variants. I think of this as a sort of blurry microscope. There is a level of specificity past which you can never focus because of that true biological variability at the tips of the phylogeny. So using a particular marker sequence such as the 16S, if there is genomic diversity that occurs at below the level at which variants occur in the 16S marker, then no, you won't be able to see it with amplicon sequencing. On the other hand, there's consistent linkage of function with phylogeny. It's diverse but not random, such that down to that level of blurriness, you do get a fairly accurate profile of what the bugs are probably doing.
Our next question is from Sanjita Khare of the NCTR FDA. What is the best strategy to correlate microbiome with immune response mucosa-associated microbes, stool microbiome, etc.? If mucosa associate, which site, ileum, colon, etc.?
Great. If I interpret that correctly, there's sort of two questions linked in there. One being the methods by which you can associate microbes with phenotype or environment, and the other being sort of detailed biogeographical variation in the gut itself along the colon, for example. So for the first question, there's a lot of great work in biostatistics and computational research right now in ways of handling microbiome phenotype associations, very much the same way that, say, GWAS associates genetic variants with phenotypes. Or several years ago, microarrays helped to associate transcription with phenotypes. The methods are often very similar. There are tools such as Lemma for microarrays, EDGER or DE-seq for RNA-seq data, GATK for genetics data, that all perform the same kind of fancy t-tests. And there are packages out there that will do the same thing for microbiome data. We have two referred to as Lefsa and Maslin from our lab that perform univariate and multivariate tests respectively. And there are several others from other groups that all do variants on statistical tests appropriate for microbial data. On the other side of the question, as far as gut biogeography, We and others have performed some fairly detailed studies of how stool reflects different mucosal microbial communities going along the length of the GI tract. What we found, to use one example, in a primate model, is that stool very well reflects the colon mucosal community with a few quantifiable differences. So what we saw there is that overall data diversity, like we saw in the IBD example in this talk, between stool and mucosal biopsies can be quite high, but it's often driven by changes in just a few taxa that are much more abundant at the mucosa or in stool. And once you normalize those bugs, stool really does well reflect what's going on with the mucosal surface. It's true in the colon. If you move up to the small intestine, there's still a high correlation between the two, but the linkage is much less direct, and there's more noise. There are more differences driven by differences in biology, pH, and microbial density, for example, between those two environments. Whereas stool along the length of the colon, both luminal contents and stool proper, pretty well reflect what's happening at the colonic mucosa with those few quantifiable differences.
Well, thank you, Dr. Huttenhower. It looks like that's all the time we have today, and so that was our last question. First of all, I'd like to thank Dr. Huttenhower and our audience for attending today. Today's webcast will be available for on-demand viewing through February 2016. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's event. Meanwhile, thanks again for coming. We'll see you next time. Goodbye.